Okay, so welcome back to this next video in which we are discussing the mammalian circadian timing system. Okay, right, so we're in the process of discussing the structure of Krebs alpha proteins. Okay, so we've discussed that at the carboxylic acid terminus of the protein, what you have is this domain called the leucine zipper domain, which has leucine residues every 7th amino acid, and this is the site where two Krebs proteins will dimerize together. Okay, the two leucine zippers will become, they'll stand parallelly to one another. Okay, and then the leucines that project off these alpha helices will interact with one another, and this will hold the two Krebs proteins together. Okay, right, so we have one final domain to talk about now, which is this domain in between Q2 and also the leucine zipper domain there, the domain that I've now highlighted in red here. Okay, and this portion is the DNA binding domain. Okay, and within this DNA binding domain you have a large number of basic amino acids. Okay, so examples of basic amino acids are arginine and lysine. So let me show you the two main examples of basic amino acids which can be found in this DNA binding domain. So we'll start with arginine here. Okay, so I'll draw you an arginine residue. Here's the amino group, here's the alpha carbon with a hydrogen coming off it, and here is the carboxylic acid group. Okay, and now the R group of arginine consists of 3 methylene groups. Okay, and to draw 3 methylene groups, what I'll do is draw one methylene group, put brackets around that, and then subscript it 3. Then off the 3rd methylene group, you'll have a nitrogen atom with a hydrogen coming off, then a carbon atom. This carbon atom will then have an amino group, and it will also have a double bond to a nitrogen atom, which has then got a hydrogen coming off. Okay, so this is a basic residue because this nitrogen here that is double bound to this carbon, this has a lone pair of electrons. Okay, so I'll colour in this lone pair of electrons here. So this lone pair of electrons is a centre of negative charge which can interact with a free proton basically that's in solution. So what can happen is a free proton can come out of solution and bind to this lone pair of electrons on this nitrogen atom. Now the proton will have a positive charge, so that gives the end of this amino acid a positive charge. Okay, so this is a basic amino acid. Okay, because it's capable of receiving protons from solution. Now, what's the significance of having a large number of basic residues within the DNA binding domain? Well, DNA has a negative charge because of the phosphate groups in the sugar phosphate backbone. Okay, so having positively charged residues because they've been protonated, because they are basic, is an advantage for binding to DNA. The other example then of a key basic residue that you find a lot of in this DNA binding domain is lysine residues. Okay, so I'll draw the structure of a lysine residue. So here's the core structure. Then you have 4 methylene groups. So here is one methylene group, and now we want 4 of those, so I'll bracket it and subscript it 4, and then we want an amino group right on the end. Okay, now once again this nitrogen atom in this amino group has a lone pair of electrons. Okay, here. And this lone pair of electrons can accept a proton from solution, thereby giving this amino group a positive charge basically. Okay, so this is another example of a basic residue because it can accept protons out of solution. That's the definition of a base, and the significance of it is that it means that many of the residues in this basic region of the protein will have positive charge because they have accepted protons out of the solution, and these positive charges will allow them to interact with the negatively charged DNA. Okay, right, so that now is the structure of a Krebs protein. So the key portions then are these dimerization portion, this dimerization portion here with this basic portion, and together there's a name for both the basic portion with the leucine zipper. It's known as a B-zip domain. So when you have this basic portion in front of a leucine zipper like this, it's called a B-zip domain, and Krebs transcription factors are known as B-zip transcription factors because they have one of these B-zip domains, and it stands for basic region and then leucine zipper. So the zip portion is for leucine zipper. So we have a basic portion followed by a leucine zipper portion. Okay, and that's what you have at the carboxylic acid terminus of all three of our Krebs proteins that we're going to discuss. The other important domain that all of the Krebs proteins have is one of these kinase-inducible domains. At least all of the Krebs proteins we're discussing, Krebs alpha, Krebs beta, and Krebs delta. And this has a serine 133, which is going to be phosphorylated by our calcium calmodulin-dependent kinase 2 enzyme. Okay, right. So, let's firstly discuss then how Krebs proteins bind DNA. Okay, so the first thing to say is that two Krebs proteins will dimerize by binding their leucine zippers to one another. Okay, then what will happen is the DNA binding domains here in red will then bind to the DNA. Okay, and let me show you how this works. Basically what happens is the DNA sits in between those two DNA binding domains. So the leucine zipper domains here will be dimerized together, and then the rest of the Krebs proteins are not bound together, so there's a gap in between them. And what's going to happen is the DNA is going to sliver down the middle of that gap between the two. Okay, so the transcription factor is going to have the DNA sitting in between these two DNA binding domains, and then you've got the final portions of the Krebs proteins out here, the portions after the, well, in front of the B-zip domain, which are then going to be regulated by the kinase enzyme. Okay, right, so let's get another piece of paper. So what's going to happen then is these Krebs dimers are going to bind to the promoter regions of the period proteins, so period 1, period 2, and also the cryptochrome proteins such as cryptochrome 1 and cryptochrome 2. And they will bind initially in this inactive state, and they will not initially activate transcription of the downstream gene. Okay, so here we have our period/cryptochrome gene, okay, which I will highlight in green here. Okay, and what's going to happen is our dimer of Krebs proteins is going to come and bind to the promoter region of our period/cryptochrome gene. So here is our promoter region, which I'll highlight in pink here. And what's going to happen is we're going to get our Krebs dimer binding to this promoter region. Okay, so how should I show this? So we'll have initially the two leucine zipper portions dimerized together, and then what's going to happen is one is going to come out in front of the DNA helix here with its DNA binding domain, the other will go behind the DNA, okay, and then you'll have the other portion sticking out under here, and then the portion in front of the B-zip domain of this one that comes in front of the DNA double helix here. Okay, so let me colour this in. So here is our DNA binding domain of one of those Krebs proteins. The other DNA binding domain goes behind the DNA double helix. And then you've also got the leucine zipper portions in green up here. Okay, and then the portions after that, which contain, as far as we're concerned, only the kinase-inducible domain, that's the only bit that we care about, is sticking up down here. So, the Krebs dimers bind to these promoter regions but don't activate the transcription yet. What has to happen is, firstly, calcium calmodulin-dependent kinase 2 enzymes have to come into the nucleus and now phosphorylate the serine 133s on the kinase-inducible domain portions of both of these Krebs proteins. So both of them are going to get phosphorylated at serine 133. Okay, and once serine 133 has been phosphorylated, what can then happen is another protein can come and bind on top of that kinase-inducible domain. Okay, and one protein will bind to each of the two Krebs proteins within the Krebs dimer. And this protein that's going to come and bind on top of the kinase-inducible domain of the Krebs protein is called the, not the kinase-inducible domain, it's called the Krebs binding protein. Okay, and for short, the Krebs binding protein is often abbreviated to CBP for short. So C for Krebs, B for binding, and then P for protein. Okay, so 2 Krebs binding proteins therefore overall bind to each of our Krebs transcription factor dimers. Okay, and now once the Krebs binding protein is bound, that will then activate the transcription of the downstream gene, and therefore we will get period and cryptochrome proteins being produced. Okay, so overall, what light then is going to do is it's going to trigger the production of period and cryptochrome proteins in the suprachiasmatic nucleus, and this is how the period and cryptochrome proteins are going to become in tune with light dark cycles, basically. So period is going to go up in the day and down during night because of this control from the axon terminals of the photoreceptive ganglion cells. Okay, so if the cycle in period and cryptochrome is out of sync, what will happen is this will return it into sync, basically. Okay, right. So that's how glutamate is going to cause the suprachiasmatic nuclei neurons to have their circadian oscillators in sync with the light-dark cycle. We now want to discuss the other signaling molecule that is released by the axon terminals of these photoreceptive ganglion cells, and this is the molecule called PACAP, which I'll remind you stands for pituitary, that's the P, and then the AC is for adenylate cyclase, which is another name for adenylyl cyclase. Adenylate, adenylyl, it's the exact same thing. And then the final AP is then for activating protein. Okay, so this protein was originally named because it was found to activate adenylyl cyclase enzymes within pituitary cells, so in the anterior pituitary specifically. Okay, so there are 2 forms of PACAP. There is PACAP27, and then there is the longer PACAP38. Okay, so they come from the same protein. So what happens is you have a PACAP gene which produces a single protein, okay, here, and this is known as pre-pro PACAP. Okay, and what's then going to happen is we're going to cut up the pre-pro PACAP to release PACAP. Now, let's say here is our PACAP. Now, if you use the entire thing, you get the longer PACAP38, which consists of 38 amino acids. But if you cut slightly shorter and just use this portion, okay, so if you use only this portion that I'm now highlighting in green, you'll get the shorter PACAP27. So basically, by cutting up the pre-pro PACAP in different ways, you can either produce PACAP27 or you can produce the longer PACAP38 by adding on those extra 11 amino acids at the back, basically. So PACAP38 is just a longer version of PACAP27. It contains all the amino acids that PACAP27 does, just with an extra 11 stuck on the end. Okay, right. So both of these molecules, both PACAP27 and PACAP38, are going to activate receptors on the suprachiasmatic nucleus neurons. Okay, so what is the main receptor that the suprachiasmatic nuclei neurons have on their surface for PACAP? Okay, so it is a receptor known as PAC1. Okay, PAC1, and its name just reflects the fact that its ligands are PACAP27 and PACAP38, so both of these will bind to PAC1. Okay, right. So, the receptor is a G protein coupled receptor. It has these characteristic 7 membrane spanning alpha helices. So it's a G protein coupled receptor, a GPCR for short. Okay, so it has 7 membrane spanning alpha helices, and it's going to interact with heterotrimeric G proteins once its ligand, which is either PACAP27 or PACAP38, has bound to it. Now, which heterotrimeric G proteins does it then interact with? Well, it interacts with GS heterotrimeric G proteins. So what does that mean? Well, let me draw you a heterotrimeric G protein. Okay, so heterotrimeric G proteins have 3 subunits. Okay, they have an alpha subunit, and then they also have a beta and a gamma subunit. Okay, now the alpha subunit is the most important. The alpha subunit can be in two states. It can be in an on state, in which it has guanosine triphosphate GTP bound to it, and it can also be in the off state, where it has guanosine diphosphate GDP bound to it. Okay, let's say initially that our alpha subunit of our heterotrimeric G protein is in the off state and has guanosine diphosphate bound to it. So here I'm now colouring in red the alpha subunit of our heterotrimeric G protein. Now, another important thing to say is that the alpha subunits of heterotrimeric G proteins are not actually integral membrane proteins. They're not sitting in the cell membrane, but they are attached to the cell membrane. So they have this little moiety sticking off them. So what happens is onto the polypeptide of the alpha subunit, because it's a single polypeptide, okay, onto the polypeptide you attach onto one of the amino acids a lipid moiety, basically. Okay, you attach a really long chain carboxylic acid onto the side of the alpha subunit. Now usually the long chain carboxylic acid that you attach is something known as palmitic acid. Okay, and I'll show you this. Palmitic acid is the old name for a molecule which would now be called hexadecanoic acid. Okay, and I'll show you the molecular formula for hexadecanoic acid. So here is the carboxylic acid group here. Okay, that's the first carbon. Now, hexadecanoic acid, this name tells us exactly what we're dealing with here. It tells us that we're dealing with a 16-carbon, fully saturated carboxylic acid. So we need 16 carbons. We've got 1 here, so we need another 15. So 14 of those are going to be in the form of methylene groups. So I'll draw a methylene group here. And now, because I don't want to have to draw 14 methylene groups, I'll draw 1. And then bracket it like so, and then subscript that those brackets 14. And what this means is repeat this methylene group 14 times. And then finally, off the final methylene group, you then have a methyl group, like so. And this takes us up to 16 carbons. So we have 1, 2, and then the 14 in the middle, which takes us up to 16. Okay, so this is the structure of hexadecanoic acid. So, what you can do is you can attach these molecules onto the amino acid residues within the alpha subunit's polypeptide. Specifically, they get attached onto cysteine residues within the alpha subunit's polypeptide. Now, every alpha subunit gets 1 palmitoyl group added onto it. Okay, some alpha subunits even get an extra lipid moiety stuck on, which is different type of long-chain carboxylic acid. Instead, it's something called myristic acid. Okay, and myristic acid is the old name for a molecule that would now be called tetradecanoic acid. So a 14-carbon, fully saturated carboxylic acid. Okay, right, so the same thing, just with 14 carbons rather than 16, so reduce this 14 here to 12, and you've then got myristic acid. So some alpha subunits get 2 lipid moieties attached to them. Now the fundamental idea here is that once you've got this lipid moiety dangling off the side, this is extremely hydrophobic. It doesn't interact well with water. So instead what happens is it implants itself into the hydrophobic core of the phospholipid bilayer, and that's going to hold the alpha subunit attached to the underside of the phospholipid bilayer, basically. Okay, right, so there we go, that's the alpha subunit here. Okay, next up, we want to discuss the other 2 subunits of the heterotrimeric G protein. So you're also going to have a beta subunit and a gamma subunit, and these under no physiological circumstance ever come apart, so they're often collectively referred to as the beta-gamma complex, but they are fundamentally 2 separate polypeptides. So in blue here we have the beta subunit, okay, and then in green we have the gamma subunit. Okay, so you attach a beta subunit with a gamma subunit to make the beta-gamma complex. The gamma subunit also has a lipid moiety attached to it, okay, so a really hydrophobic molecule that sticks off one of its amino acids and implants into the phospholipid bilayer. Now this is a slightly more complicated lipid structure than palmitic acid or myristic acid. It's something known as geranyl group. Okay, but the principle is the same, that you've got a very hydrophobic structure sticking off the side of you, and this will implant into the phospholipid bilayer and holds the beta-gamma complex at the underside of the phospholipid bilayer. Okay, now, what then happens is when the alpha subunit is in the off state, it will associate with a beta-gamma complex, and now we have the full heterotrimeric G protein. Okay, right. So, next up, we want to discuss the specific heterotrimeric G proteins that the PAC1 receptor is going to interact with once it's got its ligand bound. Okay, so the PAC1 receptor is going to interact with GS heterotrimeric G proteins. Now, what does that mean? Well, heterotrimeric G proteins are named not according to which beta and which gamma subunits they have, and there are multiple beta and gamma subunits. There's 6 different beta subunits and 12 different gamma subunits, okay, but we don't care what those are, okay, it's just a beta and a gamma subunit as far as we're concerned. Heterotrimeric G proteins are labeled according to which alpha subunit they have, and there's 21 different alpha subunits, okay, so labeling up a heterotrimeric G protein as GS tells us that the alpha subunit is an alpha S subunit. Now there are multiple splice variants of the G alpha S gene, okay, but they all pretty much do the same thing, so we won't go into that, okay. It's just one of these alpha S proteins, but be aware that there are multiple G alpha S proteins because the G alpha S gene has multiple splice variants, but they all pretty much do the same thing. Okay, right, so here we are, we're set up with our heterotrimeric G protein here and our G protein coupled receptor PAC1 here. What's then going to happen is the PACAP27 or PACAP38 is going to come along and bind to the PAC1 receptor. PAC1 receptor will then change conformation, okay, and will now be able to bind to the alpha subunit of a GS heterotrimeric G protein, which is an alpha S subunit. Now, let's just discuss a few little portions, well, a little bit of nomenclature for the different portions of a G protein coupled receptor. Okay, so, in vivid purple here, I am shading the 7 membrane-spanning alpha helices. Okay, so you have 7 of these, and they're labeled up transmembrane alpha helix 1, TM1, which is the first one, then TM2, which is the second membrane-spanning alpha helix, TM3, which is the third membrane-spanning alpha helix, TM4, the fourth membrane-spanning alpha helix, TM5, the fifth membrane-spanning alpha helix, TM6, the sixth membrane-spanning alpha helix, and then TM7, the seventh membrane-spanning alpha helix. Okay, then other portions that are given names, we have these 3 intracellular loops here, which I'm colouring in orange. So the loop between TM1 and TM2, the loop between TM3 and TM4, and the loop between TM5 and TM6. Okay, these are known as the intracellular loops. Okay, and they're abbreviated to ICL. So I for intra, C for cellular, okay, and then L for loop. So we have these 3 intracellular loops which are called ICL1, which is the first one between TM1 and TM2. Then we have the second one between TM3 and TM4, ICL2, and then the third one between TM5 and TM6, ICL3. Okay, right. Then we have also extracellular loops. Okay, 1, 2, 3. So we have, these are on the outside of the membrane, clearly, and those are labelled up ECLs. So we have the first extracellular loop, ECL1, then we have the second extracellular loop, ECL2, and then the third extracellular loop, ECL3. Finally, these portions in front of the first membrane-spanning alpha helix and after the seventh membrane-spanning alpha helix are also given names. So this portion prior to the first membrane-spanning alpha helix, this is known as the amino terminal domain. Okay, and that's often abbreviated to ATD. So I'll write this down here where I've got space. This stands for the amino, and then the T is for terminal, and then the D is for domain, so amino terminal domain. And then the portion that's after TM7 down here, which I'll highlight in green, this is called the carboxy terminal domain, or just the C terminal domain. Okay, and the C terminal domain is usually abbreviated to the CTD. Okay, so this is also called CTD. Right, so when the ligand binds to the PAC1 receptor, what will happen is you'll get a conformational change in the PAC1 receptor, and this will cause a conformational change on these 3 intracellular loops. So these will change conformation, and they'll make available a binding site for the alpha S subunit of a heterotrimeric G protein. Okay, and we'll continue this discussion in the next video.